think here, I can get to this. That's the best we can do. Yeah. Okay, sorry about that. Good thing we started early. Anyway, back to my title slide. ChatGPT gave me a lot of options that I wasn't really happy with. And so finally, I decided to use the photo of the sunset I took last night here. It was a spectacular one. I hope you all got to see it. If not, there'll be a repeat tonight, I'm sure. So I am a bioengineer. I do roughly 80% experimental work, 20% modeling, but if you integrate that 20% over many, many years, I've worked on quite a few mathematical modeling projects over the course of the career. So what I thought I'd do today is give you sort of an overview of different kinds of modeling projects that we've worked on, mostly focused on the tumor microenvironment. So we're very interested in this tumor microenvironment and how it affects tumor progression and treatment responses. And just conceptually, the tumor grows in a confined space. It produces mechanical forces, solid stresses that disrupt the normal tissue architecture. That tissue damage causes inflammation and infiltration of normal cells and immune cells that are trying to address that tissue damage, basically. And so the result of that is deposition of additional matrix, stiffening of the tissue. Those cells are also producing cytokines and growth factors that do things like recruit new blood vessels, but also make existing blood vessels more leaky. And so you get this change in fluid homeostasis in the tissue. So you get leakage, high interstitial fluid pressure, and the lack of lymphatic vessels in the tumor also means you don't have drainage. So there's a buildup of tissue pressure. And all of this has implications for tumor malignancy and how these tumors respond to treatment. So in the next 30 minutes or so, I'll talk about some of the challenges in the tumor microenvironment that we've identified and some of the models that we've developed to try to learn more about those processes or find strategies to overcome them. So I'll first talk about this issue of fluid homeostasis. Then I'll talk about some models that we've done for cancer invasion and metastasis, and then immunotherapies, and I'll end with a short discussion about how we're measuring solid stresses in brain cancer patients. So tumor vasculature is structurally and functionally abnormal. So you can see the normal vasculature up here, tumor vessels down here. They're highly dense in some regions of the tumor don't have any vasculature, so you get this hypoxia and necrotic region that you see there. And so over the years, we've used lattice Boltzmann modeling to look at things like hemodynamics and cell-cell interactions in the blood flow. Also vascular adaptation, how the fluid forces actually change vessel diameters and cause adaptation of vasculature. And also lymphatic clearance, and that's one of the things I'll focus on here. Early in my career, we were interested in hemodynamics and how interactions between red blood cells and white blood cells can help in the immune surveillance. Basically, what we found was there are ways to enhance the aggregation of the red cells so they travel down the center of the flow, and that excludes the white blood cells, lymphocytes, and other leukocytes to the vessel wall, enhancing surveillance. And so that was something we were doing back in the 90s, trying to enhance anti-tumor immunity. And so you can imagine, no matter what we did, even if we enhanced the amount of leukocytes that were getting into the tumor, it didn't help because we didn't know about checkpoint inhibition or checkpoint pathways. So there was always immunosuppression. And so we moved on from that project. It came back a few years later, as you know. So in addition to this, the increased source of fluid when you have tumor growth because of the enhanced hypoxia and VEGF that makes vessels leaky, providing more source of fluid in the tumor, we have a lack of drainage. So we're very interested in the lymphatic system as well as the blood vasculature because that's what drains the fluid. So the lymphatic system is separate from the blood vasculature. Fluid is released from blood capillaries into the tissue that gets taken up by lymphatic capillaries, and it goes through lymph nodes and eventually through the thoracic duct back to the systemic circulation. And the key here is that these lymphatic vessels can contract to drive the flow. And what we wanted to understand is how this works in normal tissue and eventually how it works in tumor tissues and how we might be able to enhance that drainage. But we had to start with the question of what actually controls this contraction. Because in the context of the cardiovascular system, we have a central pump, the heart, and we have pacemaker signals that cause the contractions, and those contractions are mirrored downstream. In the lymphatic network, you don't have a centralized pump. Each one of these segments can contract, and the question is, how are those contractions initiated? And how are they coordinated over this network? And so we did some modeling for this while being Lee and Jim Bache helped develop these models. So the domain looks like this. We have a single vessel from entrance to exit, a number of lymphangions that are defined by these intraluminal explicit mechanical valves that are actually moved by the fluid. We have a porous media out here, so fluid can come in through this tissue boundary, into this porous media, and into this initial lymphatic. This vessel wall can contract, and it's a mechanical wall with viscoelastic springs. The fluid can move the wall with forces, and the wall can move the fluid. And that force on the wall is determined by chemical kinetics of calcium and nitric oxide. So as calcium accumulates in the cytoplasm of the muscle cell, the cell contracts. That is enhanced by stretching of the vessel through the action of stretch-activated ion channels. And this can all be tempered by nitric oxide, which blocks this calcium contraction and is produced by shear stress on the endothelium. So we have two mechanobiological pathways here that are competing to regulate these contractions. And it turns out with these two mechanisms, you can get autonomous contraction, autonomous pumping of the lymphatic vessels. We can look at pressure as well as nitric oxide. We can change the valve configuration. But to summarize what we've learned from this model, these two competing mechanical signals are sufficient to regulate the contractions and flow through these vessels. And the way that works is, so we have flow coming into this vessel, this valve opens, we create shear stress in this region, that creates nitric oxide, which relaxes the vessel locally. That relaxation is very brief. Nitric oxide has a lifetime of about 1 second. And so after that dissipates, then we're set up for another contraction at this next time point to the right. When a contraction happens there, flow goes downstream. It opens this valve and creates shear stress and nitric oxide to propagate this down the vessel. So through these oscillations of nitric oxide and calcium, we get contractions and relaxation. And so the nice thing about a model like this is you can challenge it with different perturbations that the vessels should respond to in normal physiology and see if the simulations agree with what we know. And that's what we're doing here. So when you're laying down, we have to have some constant low level of fluid clearance. And so that's shown in these autonomous contractions down here, and the red traces here for diameter and flow. What happens when we elevate the limb? When you elevate the limb, the gravity is driving the flow, and that creates shear stress that makes nitric oxide that prevents contractions. We don't want contractions when gravity can drive the flow because the contractions increase resistance to flow. And that is shown in the black trace here. We have no contractions, but non-zero flow. What happens when we stand up? Now you get a buildup of fluid pressure in the bottom of this vessel. It stretches it, initiates contractions, and now we're pumping against the direction of gravity in this blue trace. And the important thing here is that all this different emergent behavior was not due to changing things in the model to force this to happen. It only came from changing the direction of gravity in the model. So now what we're doing is making this, looking at complicated networks of lymphatic vessels, because we're interested in the contractions are coordinated between daughter branches. And to do this, we have to increase the domain size, and to keep computational efficiency reasonable, what we're doing is restricting the lattice Boltzmann calculations to the area just around the vessels and in the vessel. And then we use FDM calculations in the porous media outside here in the larger domain. So next, let's talk about invasion and metastasis. So we've developed a number of agent-based models over the years to look at questions of tissue mixing and cancer cell migration. But the model I'll talk about here is motivated by images like you see here. What you see is cancer cells proliferating and moving and impinging on the surrounding tissue. But the thing that drew my attention was these brown stains. That's collagen 4 basement membrane. So basement membranes are the structures that contain tissue, organize structures, and define different organelles or organs in the tissue. So what I see here is it's a competition between the cancer that's growing clumsily and the host immune and stromal cells that are trying to contain that tumor. So there's this battle between tissue repair and cancer growth. So we developed a model to try to look at these issues. And just to summarize, this is an agent-based 3D model, and the cancer grows, invades, and can digest basement membrane structures. The epithelial and endothelial compartments, their rules are to try to maintain their two-dimensional structure. So if they get a hole, they try to reseal that hole. So the underlying rules here are to try to decrease the topology of the system, or to maintain the topology. So we can look at validating this model by showing that it can reproduce wound healing, because that's sort of the way the agent-based rules were developed. And so here we have ZY plane and XY plane in the 3D model. We have a blood vessel, skin interface. So the yellow is epithelium, the green is endothelium. And then we have a gland down here that has epithelial cells. And what we do then is introduce a wound, and then run and see that clot dissolves and the topology to an Euler number that departs from 0 as that clot is being healed, and then returns to 0, showing that the topology had turned to normal. And so you can see the reconstitution of the endothelium, basement membrane, and epithelial sheets. So the cool thing about this, I won't go through all the simulations. We can create an array of gland structures with blood vessels in between. We can seed a cancer cell here, and changing different model parameters, we can create different patterns with compartments that have different topologies. And again, we can measure those, and we can compare those with what we see in images like this, and learn about what are the mechanisms that might have been responsible for creating this tumor. So next, I want to talk about immunotherapy. As we all know, immunotherapy is a very hot topic. It's doing, it's creating miracles in some patients, but it's very inconsistent. And this inconsistency is what we're interested in. We have system immunology models where we can look at things like distribution, T cell activation, and how these processes affect tumor regression. And this was motivated by data such as you see here. So in melanoma patients and breast cancer patients, some who get anti-PD-1 therapy, you can have these dramatic responses in some patients, but in other patients, there's little or no response to the therapy. And you can maybe explain this by being due to differences in patient immune systems, maybe differences in the tumors themselves, maybe differences in diet and microbiome. But it's more difficult to explain data like this that came from mouse models of breast cancer. So again, with anti-PD-1 treatment in the blue lines here, some mice are cured, the tumors go away. Other mice don't respond, and the tumors grow like controls. And this is the case even though these mice are genetically identical. They have identical tumors implanted, and they have identical diets. So the question is, what's responsible for why some of these respond and others don't? So we come back to this cycle of anti-tumor immunity. So primary tumor releases antigen, it gets into the lymphatic system, the antigen accumulates in lymph nodes, and at the same time, naive T cells are circulating through the blood circulation. And we have about 10 to the 11th different clones of NT cells in the body. And the key here is that only a small fraction of those, a handful of those, can recognize tumor antigen. So it's a potential rare lymph node trying to find antigen that matches their T cell receptor. If they find a match, they proliferate, and there are 500 of these lymph nodes, by the way. So they go one at a time. And if they find a match, they proliferate, or activated proliferate, and then they go back to the systemic circulation looking for the source of the antigen. And if they can find the tumor in this case, they can come out and start to kill it. So we look at this with a PK type model where we have compartments representing each organ. The compartments are connected anatomically by the arterial and venous systems, as well as lymphatics. And in this lymphatic network, we have the lymph nodes where activation can happen. And then we can so this model, it's a shame you can't see the top of these slides. So anyway, maybe I can move this out. That's better. More, we can get rid of it. Hide video panel. Oh. Hide floating menu. There we go. Should have done that a long time ago. So we have a stochastic part, and that's the part where the naive T cells are circulating. They go from lymph node to lymph node. We have transit times and residence times in each organ and lymph node. We check to see if it's in a location where the antigen concentration is high enough. And then if it is, we activate that T cell. It becomes a deterministic model at that stage. We have a lot of effector T cells now that have proliferated. They go to the circulation, find the tumor, and start to kill cancer cells. So if you look at systemically at where the antigen distributes, of course, the tumor has the highest level. And in the red and blue here, we're just showing two different antigen production rates. Other organs get very low levels of antigen through the bloodstream. Plasma has moderate levels. In terms of the lymph nodes down here, of course, the tumor draining lymph node gets the highest level of antigen, but some does accumulate in other lymph nodes. To walk you through how the simulations look, the tumor is growing up here. It's releasing antigen that gets taken up mostly by this tumor draining lymph node, but other lymph nodes as well. At the same time, we keep track of the location of these naive T cells. So for example, this T cell visits the intestinal node first, and then the lung, and then the head and neck, before eventually by day 6, it makes its way to the tumor draining lymph node. And at that point, we check antigen level, and we have a putative threshold here above which activation. So it's point, the antigen level in that lymph node is high enough, so we activate that T cell. It proliferates in the lymph node, is released and finds the tumor and starts to kill cancer cells. So that's how, in general, it works. Let's look at two of the important parameters in the model. One, of course, is this antigen production rate. If you have low antigen production, we don't see any activation of T cells. If we have higher antigen production, we start to see these stochastic events where the T cells are activated and regress the tumor. Down here is the lymph node fractional flow rate. So that's the other important parameter. And it basically describes of all of the naive T cells that enter the vasculature of an organ, what fraction of those actually go through the parallel circulation of the lymph node. So you can imagine that's a pretty small number because the volume, if you just base it on blood volumetric flow rates, the volume of the lymph nodes is much smaller than the volume of the organ. So, but increasing that parameter, you can see leads to more and more of these activation events. So because the antigen production rate is so important here, there are, of course, ways that people are trying to enhance antigen production through things like chemotherapy or heat treatment or radiation therapy. So we decided to look at how radiation might affect these responses. And so over here, we have low antigen production. So with no radiation, we don't see any activation in these tumors. If we irradiate the tumor, now we're releasing more antigen from the tumor, and we start to see activation of these naive T cells. And so we get an effect of tumor killing by the radiation itself in these sharp declines. But we also have a component that comes from the anti-tumor immune response from those T cells down here. If we irradiate the tumor draining lymph node along with the tumor, so things change a little bit because now we're actually killing some of the T cells that are proliferating in that tumor draining lymph node. And so that actually decreases the response rate a little bit. For the high antigen production case, it's a little different. So we have plenty of antigen here to activate those naive T cells. If we irradiate the tumor, we actually remove some of the source of antigen by killing those cancer cells. So we see a slightly lower response rate when we do that. So it really depends on the baseline antigen conditions in the tumor, how this radiation therapy is going to work.
Tell us what kind of radiation therapy you perform there? What is the dose of radiation? Did you apply that radiation to the lymph node as well?
It was the same dose to the, yeah, I think it was 5 gray over, you can see distributed over those 35 days, starting at day 14. I can get you the details, it's in the paper. So I mentioned that these T cells are activated in the lymph node. They go into the blood circulation and have to get to the tumor. And this is another critical step in immunity because we need to get those T cells distributed in high numbers throughout the tumor. And so we've done some detailed spatial modeling of that process. And Mohammed Nigmaneshi is leading this effort. And he is actually looking for a job if anybody has any openings. But here we have a tumor that's seated at the center of the domain. It grows. It can become hypoxic. It produces VEGF. It recruits these angiogenic blood vessels that are guided by the VEGF gradient. So this is a pretty realistic vascularized tumor, and we can use this then to look at T cell infiltration through those vessels. So the model is a hybrid model. We have the cancer cells and endothelial cells on the lattice. We do FDM calculations for things like oxygen and growth factors and drugs if we inject them. Here's what it looks like for an untreated tumor from the side view and top view. Again, we keep track of metabolism in the tumor so we can know where necrotic, quiescent, and proliferative cancer cells are. And we've used this model to compare different kinds of treatments like maximum tolerated dose versus metronomic versus combination anti-angiogenic therapy. I don't have time to talk about that, but using the same kind of model, now we can introduce T cells into the vasculature and ask questions about this issue of cold versus hot tumors. So hot tumors defined by one that has T cells that are well distributed, and all the cancer cells are pretty accessible to the cytotoxic cells. And some of the key parameters in this model deal with how these T cells are rolling, adhering, and extravasating from the vessel, what they do when they get out of the vessel, how well can they move through the matrix and counter these cancer cells. So this is just the summary. Cells can come out, conjugate with cancer cells. They can kill them or dissociate. They can become exhausted, or they can come back and kill more cancer cells. We can inject, in this case, CAR T cells into the systemic vasculature with some function, and then keep track of how many free T cells and where they are, complex T cells, exhausted T cells. And from this free T cell population, we calculate what we call a hotness factor. So this is describing how many T cells in the tumor, but also how uniformly they're distributed. So it's a distance calculation that takes into account how far away from a given cancer cell the T cells are. So the higher that hotness factor and the longer the window, the better the therapy should be. So just to quickly go through one data slide from this, increasing the rolling adhesion and firm adhesion or decreasing suppressive factors that are produced by the tumor can all enhance this hotness factor. But the thing that's most sensitive here is the collagen barriers. So once it's not enough to get the T cells into the tumor out of the blood vessel. The key step is to get the T cells to migrate as quickly as possible through the matrix. So this predicts that things like collagenase or hyaluronidase would do a good job at increasing this hotness and helping therapy. So in the last few minutes, I'd like to talk about what we're doing with solid stresses and how we're making measurements in actual patients. This is GBM growing in the left hemisphere of this mouse. And you can't see the tumor, but you can see the effect that the mechanical forces it's producing have on the normal vascular structures. So this deformation is due to this buildup of solid stress, and that stress can cause neuronal apoptosis in this brown stain here. So you end up with a decreased neuronal density around the margin of the tumor due to that death. So this is an important clinical phenomenon. Over the years, we found ways to measure solid stresses in mouse models. And what you see here is a tumor from a mouse that's been taken out. If you cut through it, it actually deforms. That it's deforming because you're releasing built-up solid stresses in the tissue. And so you can use that phenomenon to actually make measurements of the solid stress. So what we do is embed it in agarose, make the cut, and then do high-resolution ultrasound imaging of that interface to get the topography after the relaxation has happened. And then from that topography, you can use mechanical models to back calculate how much stress had to be in the tissue when the cut was made to result in that deformation. And so we've done that in mouse models for many years, but now we've started doing it in patients. And so we're collaborating with a couple of surgeons who are willing to help us with this. They use the Brain Lab neuronavigation probe. And after they make a craniotomy and before the surgical resection of the tumor, they map out the surface of that craniotomy brain tissue. Here we have 16 different locations, so we get the swelling after the pressure is released. And we can use that measurement in conjunction with time-resolved MRI images of the tumor as it has grown. to estimate not just solid stress, but also brain loss in the patients. So let me walk you through that. So we have the tumor size from two different MRIs. We have this measurement of the tissue relaxation from the solid stress. In the two extremes, cases that are shown here, in this extreme, you can imagine the tumor grew to a large size, but there's no bulge after the craniotomy. And what that would indicate is that the tumor has not displaced the tissue by solid stress, but it's actually replaced the tissue. So you've had brain loss in this case. In the other extreme, if you go from a small size to a large size and there's a corresponding large bulge, now you can say that the solid stress has caused that bulge and you have displacement instead of replacement. So using this kind of logic and some curve fitting here, what we've estimated is that in these 14 GBM patients that we've looked at so far, there's an average of around 4% brain loss up to 10%, and it's higher in the primary GBMs than in brain mets. So I'll end there. I think we're a little early. That's good. By acknowledging all these people that worked on these various models. And I'd be happy to take any questions.
Okay, questions? Anyone?
Hi, sorry, nice talk. I was curious about the last project you mentioned about the internal pressure of the tumors. You told us that you measured that in glioblastoma, correct? Or what was the tumor? Was a brain tumor?
These are GBMs, yes.
Yeah. Are you planning to measure that also in other tumor types to see like how it changes between different solid tumors?
Yeah, I mean, over the years, we've thought about different ways to try to do that. It's an invasive process usually because you have to take a piece of the tumor out and then measure some deformation because you have to release some solid stress. So we've thought about ways to do it with biopsies and also, probes that you could potentially insert that would have piezoelectric circuitry to measure the internal stresses. But we haven't really done it in any tumors yet besides the brain, because this was sort of it's a fortunate but unfortunate thing that when the craniotomy is done, you get some relaxation of that solid stress naturally. So it's already there. The surgeon doesn't have to do anything special. We don't have to get any additional IRBs to do this.
I was wondering if you have any idea why the liver has such a low antigen load, even though so much of the body's blood goes through the liver all the time. I thought that was interesting that the liver seemed to have almost negligible amounts.
Yeah, I think in the model it's because we have a pretty high clearance rate for proteins in the liver. So things are being dissipated in the liver, even antigens, right? So it's filtering things out and they're degrading.
A question about the modeling of the tumor stroma patterning. Can your models incorporate the dynamic stroma? Because oftentimes during treatment, you see a dramatic changes in the sort of tumor stroma architecture. And this sort of related question, how does it impact lymphatic vasculature and blood vasculature apart from mechanical stress? Or do you see a connection between the specific patterns of stroma and the messiness in the vasculature, both lymphatic and blood vasculature?
I mean, in general, radiation is stiffening tissue, causing, matrix deposition and things that does affect things like blood vessel tone. And we actually have a project going on right now with Tim Padera's group where they're irradiating lymphatic vessels, and it does stop the lymphatic pumping. And so we're trying to figure out whether that's because just stiffening of the tissue or whether it's actually damaging the muscle cells in some way. And we don't have the answer yet for that. But definitely, the radiation induces inflammation and stiffening, and all of that changes mechanobiology of many different cells, right?
We have 2 more minutes, so I will ask my question. So on the GBM example, of course, I have a question there. So I was interested in the herniation effect when you do the craniotomies and measuring that. Have you tried to cross-correlate that with either MR elastography measures of in vivo understanding of what we think the MRI is telling us about elastography with these tumors? And have you connected it at all to like imaging patterns of edema?
We have not done those things, but I would love to talk to you about some of those things.
Yeah.
What we're doing now is collecting blood and tissue from these patients and trying to do some transcriptomics at the same time that we have some measure of solid stress. But yeah, I think we can have some conversations.
Yeah, I think we have some overlapping data that might be useful to
Great.
help in this space or be, collaborative.
Yeah.
Okay, well, let's thank Munn once again for a very nice presentation.